Okay.
Hi friends, today we are going to see about B cells or B lymphocyte cells. B cells are formed within the bone marrow. It's a type of lymphoid cell, the humoral immunity of adaptive immune system. But the outer coverage of B cells, which are consist of B cell receptor. These B cell receptors, which is used to bind a specific antigen for the B cells. Then we are going to see about function of B cells. This function of B cells, which is different types of B cells produced by a human body. First one, the performs the role of immune surveillance, and the second one is BCR on its surface, which is helps in binding with one particular antigen.
Okay. Now.
Okay. And now the B cells become a plasma cell which produce antibody at the time of immune response. Then the B cells circulated as a memory cells also, and etc. Now we are going to see about stages in B cells. The stages in B cells are maturation, activation, and differentiation. First, we are going to see about maturation. Maturation, it's one of the maturation stage of a B cell. Active B cell become the plasma cell which produce antibodies. When the B cells become a matured state, activated B cells are make a mature state. At the time, these B cells are become a plasma cell, and which also produce antibodies. Then we are going to see about activation stage. In the active stage, the B cells are B lymphocyte are activation is just as a clonal proliferation and terminal differentiation into plasma cells.
Okay.
And now the T cell dependence activation. We are going to see about the T cell dependent activation. Here, the B cells, which is triggered with it and encountered matches antigen. Antigen will come and triggered with the B cells. And now this B cell engulfs antigen and digests it. Now these antigen was digested inside the B cells, and then these display antigen fragments bounded in the unique MHC complex molecules. The MHC means major histocompatibility complex. Now the compactation of the antigen and the MHC attracts the help of maturation and matching the T cells. Then here after this attraction, this the cytokine which is secreted by the T cells and help the B cells to multiply and mature into a antibody production and producing plasma cells. There, this antibody binded with antigen in the blood cells and the complex cleared by the complement by the liver or spleen. Then these T cell dependent, which is consist of antigen, must contain both B and T cell epitopes. Here we can see this is an antigen which consists of T cell epitope and B cell epitope.
Okay.
And now we are going to see about T cell independent activation. In this T cell independent activation, which consists of two types. There are two, type 1 and type 2. This type 1 and type 2 are both are T cell independent, but the type 1 type 1 is which is also called as polyclonal. Now we are going to see about type 1. The B cell binds to a specific antigen and receives another secondary activation by toll-like receptor. The toll-like receptors are like TL4 for LPS and TLR9 for DNA. These activated B cells which are restricted to IgM antibodies. Now we are going to see about type 2. The type 2 which is occurs when the antigen that are expressed on the surface of the pathogens B cell activated takes place. Many bacteria has repeated carbohydrate epitopes that simulates B cell to cross-link their IgM antigen receptor, and this type 2 activation which leads IgM synthesis in the absence of T cell stimulation. Here the conjugated vaccines are made to provide a stronger immune response against the foreign molecules.
Okay.
And now the last stage of B cell, which is a differentiation. In this differentiation, B lymphocyte express immunoglobulin on their surface as a clonal receptor, but they do not secrete antibodies. B cells stimulates with the mitogen proliferation and differentiation into plasma cells. From there, each of which produce thousands of antibody per second. And we are seeing this diagram as this is an B lymphocytes, and the antigen are foreign bodies which is get get inside this B lymphocyte, and which is activated to form plasma cells, and from these plasma cells the antibodies which are produced, thousands of antibodies which are produced. Thank you for seeing this video.